well, I can just start while we're opening the slides. But yeah, I'm really, really excited to be here. It's always nice to be in a room full of experts. So I'm going to use that actually and grab as many of you as possible later to discuss some of the species we're working on. Yeah, so I'm Aline. I'm a conservation scientist here at the Botanics, and I'm going to talk about how we use conservation translocations to help our Scottish plant recovery. And I'm going to use the opportunity to just really briefly introduce the wider Scottish Plant Recovery Programme, which is a core programme here at RBGE. And also we've just received recently a 3-year Scottish Government's Nature Restoration Fund, difficult to say, and where we're going to focus on 10 key priority Scottish plant species that we're going to safeguard in large genetically diverse ex situ collections, but also translocate back out into the wild. And you see the 10 species there. It's 5 tree species and 5 herbaceous species. And if you have any particular interest in any of these species, please do get in touch because we're working on these species really intensively over the next few years. And I've been asked specifically to give an update on the Alpine Blue-sow-thistle. It's a Cerbata alpina, which is excellent because it's a really, really good example of the type of work we do and the type of work that is required to help threatened species through translocations. And this is Cerbata alpina. It's a very pretty plant, which also helps when promoting plant conservation. And it's about 1.5 meters tall. It's a perennial plant. It's insect pollinated. The seeds are wind dispersed. And it's fairly common throughout Scandinavia and through European mountain ranges. But in the UK, and that's us there, it's really rare with only 4 populations that remain. Now, all in Scotland, all within Cairngorms National Park. And it's not just the number of populations, it's also the size of each population. So on the right here, you have a typical population in Scotland, which grows on these really steep, slimy ledges that where they escape from grazing. It's quite a palatable species, tasty to sheep and deer. And on the left, I've taken a picture in Switzerland this year where honestly, the whole forest floor is covered, like dominant. It looks a bit, it was taken late in the season, so it's not the prettiest display there, but that is all Cerbata alpina, where habitats are much more continuous. Consequently, in Scotland, on the right, you can see the recruitment isn't great. So you have seeds collected from Norway on the left compared to seeds collected in Scotland on the right. Now, this isn't exactly fair. We do find viable seeds in Scotland as well. It's just to make a point, but more often than not, the seeds look like this. So clearly not particularly viable, and we've never ever seen any seedlings and wild populations in Scotland. And quite often when we see this lack of reproduction or issues with reproduction, there are genetic issues. So we are looking into this as well. So also you've seen where the populations are. They are on very precarious ledges where one landslide could take them out. So one of our conservation measurements are conservation translocations. And conservation translocations refers to the deliberate movement and release of living organisms for conservation purposes. And on the right, you see the Scottish Code for Conservation Translocations, which we follow as it gives best guidance to do so. Now, the steps that are required, well, the steps that we take to conserve species using conservation translocations are quite a few, and the following. So it's always good to start with a genetic study because it gives us information about whether or not there might be genetic issues such as inbreeding or low diversity. It helps us determine source populations for translocations, and it helps us understand a little bit more about the reproductive biology and other things about species. Then next steps would be to go into the wild, collect seed if there are any, or rootstock, bring them back to the nursery, hand over the plants to Hoare to do all sorts of magic to them and create lots of plants, which are then ready for translocations. And that requires a lot of space, a lot of time, and a lot of expertise. Then if the genetic shows that there are issues, we can test the potential for genetic rescue, which is pretty much just increasing the fitness of populations by introducing new genes. And we can do that quite easily by doing controlled pollination experiments in the nursery and then measure fitness. Again, ideally, we do that over several generations. Requires a lot of time again. And then we can conduct our translocations. And I put trial in brackets because it's always good, and we've tried it, to test when not to plant them mainly. And of course, to understand where are particularly good to plant them. So, and then finally, really, really importantly, it's been touched on earlier, the very important monitoring phase, because otherwise we'll never understand the long-term successes and failures of our conservation measures. Right, so we've done all these 6 steps for Cervita alpina, and these are the 4 wild populations that remain. Coricanda, Cainloch, and Corifee, and Loch Nagar, as you see, very small populations in tricky locations, and we did genetic analysis using microsatellite or SSR loci, pretty much just small bits of DNA that we compared between all individuals. And the genetic data showed that we have less than 20 plants that remain at each location. 20 genetically distinct individuals, 20 genets, less than 20 genets per population. So this is good and bad news. The good news is it's more than one. It's always good to have more than one. Bad news is, in the conservation genetic context, an often quoted figure is that we should have 500 to 5,000 individuals to retain healthy population, genetically healthy populations. Now, this is not going to happen, is it? And for many other threatened species, we don't have these numbers. So the low numbers are a concern, but also the data also showed that on each ledge, we have really highly related individuals. So we have full siblings or cousins that are on these ledges. Consequently, there's a lot of inbreeding. It's a very low diversity compared to more widespread Norwegian populations, for example. There's lots of clonal growth, which is neither good nor bad. It's just what it is. And then there's no gene flow, so no exchange of pollen or seed between these populations. So they are really genetically isolated patches. Now that could explain why we have a lack of reproduction in these populations. So it's a bit of a hint that there might be genetic issues, and we could try some genetic rescue. So then we went to all 4 populations, grabbed some rootstock, 2 rootstocks from each population, I think, roughly, brought it back to the nursery, and look what Hort did with them. So even starting with very small numbers, over the years, we managed to, by dividing the plants, to create quite a large number of genetically identical plants. Now, that is not ideal for conservation, but what it does allow us is to use these plants to do some experiments without harming vulnerable wild sites. And one of these experiments was to do some a genetic rescue experiment where we tested the fitness of plants after introducing new genes. So pretty much we just cross-pollinated by rubbing flowers against each other. And we had representatives of all 4 populations, plus plants from Norway as well. We had this little pollinator exclusion tunnel, that's the name, in the central picture there to exclude pollinators. And then we crossed these plants in all sorts of combinations and variations, which was a lot of crosses. And then you've got the treatments on the top right, like we selfed them, we crossed within populations, we crossed between populations, between Scottish populations, and between Scottish populations and Norwegian plants. And that gave us a number of seedlings. And to make this really brief and short, what we found in the nursery was when we cross between populations, plants tend to grow bigger, they tend to survive longer, and they tend to flower earlier. I think that's what it was. They mature or flower earlier. So now the question is, could that be a really good conservation measure to do that in the wild? And because this has been done in the nursery, it's actually really important to see whether these positive effects last when we plant them out into really harsh Scottish environments. And we love testing our plants in Scottish harsh environments. So that's what we've done. So we've taken these seedlings and planted them out to see what happens. The grey dots on the map are the wild sites, and the purple dots, we can proudly say, are all the sites that we planted. But some of these were trial sites. And for the real translocations, we used five sites. we had 3 low altitude sites, 2 at Mar Lodge and 1 at Glenlochy. And so these sites are more similar to the habitats where we find them in Scandinavia, Switzerland, where we know they're doing really well. So it's forested habitats, low elevation, and that's the 3 sites there. And then to represent more high elevation sites where we typically find Scottish populations currently, we also picked 2 high altitude sites. And then our planting design, just quickly, I really like our planting design. So because we can divide the rootstock, what we managed to do for most plants is divide the rootstock of each plant into 5, so that we have the exact same genetic individual at each site. If that wasn't possible, and because we've done so many crosses, we could use full siblings in the different sites. So we do have the same genetic diversity or even the same genetic individuals at each planting site. So the differences we will see between sites are really down to the sites themselves and not the genetics. Now we plant pretty randomly at each site, and we had about 180 plants for each translocation site, and a fairly balanced design there in terms of the numbers for each different treatment, except for the crosses with Scottish and Norwegian plants, where we just had fewer plants, just what we had at the time. Right, just to show you a few pictures of the actual planting. So as I was saying, we did have a random planting design. So on the left, you see we've just dug up holes where in places that looked really good, and then just randomly allocated plants to these holes, and then carefully planted them and labeled each individual plant. I'm looking at, I'm not going to put them on the spot. We've got a few people in this room that have been part of the planting and know exactly what I'm talking about. No. So we did have a really, really great team for planting and helping out. And then to protect the plants from damage by voles and slugs, yes, everything like these species. We also developed like built little cages made from mouse mesh. So that's what you see at the bottom there. And then once we're done with the translocation sites, top right picture, that's what it looks like afterwards. And I'm really excited because now 2 years later, we have results. I have to say, though, these are really preliminary results. And I do realize I have this don't tweet thing there. And I realize I'm being recorded. So that's not going to work. But anyway, so it's really preliminary data. So we haven't done like proper statistical analysis on this. But I'm still really excited to talk about these results. So we have the 5 sites. I'm just going to quickly explain what, the diagram there, and then we'll compare all 5 sites. So overall, we had an 82% survival rate at this site. This is our first site at Mar Lodge. And we have our treatments at the bottom, self, within, crossed, and so on. And then I grouped plants into different growth categories. Either they were dead and no growth, or they were quite small and probably won't survive, or they were quite, medium, or they looked good, or they looked really good and flowered. So that's the rough categories just for now. And then the proportion of these categories for each treatment, pollination treatment. And what we see here pretty much is that when we cross, or the more we go to, so it's like a traffic light system. So the more red is bad, green is good. So that helps a bit to get a very quick overview. So the more we go to the right, so the more we increase the diversity, lower the inbreeding, the more green we have, the better the plants look. And particularly for, in this case, the crosses between Scotland and Norway populations, all 4. I have to say it's just 4, but still all 100% of those look really, really good. When we look at the flowering on the right, it's the same thing. So the crosses between Scotland and Norway have done proportionally really, really good. Now let's. I tried to squeeze all sites onto one slide. Let's see, but it didn't quite work. So this is the same as we just looked at. I'm just gonna go. So because maybe that was just coincidence, right? So just one side. So let's have a look at the second side at Mar Lodge. And it looks exactly the same as if I had made it up, but I haven't, I promise. We had an absolutely random planting design. And look at that. So again, when we cross between populations, there is a massive increase in, well, there's an increase in green, and particularly, again, the crosses with Norway are particularly good. Maybe it's just Mar Lodge. So have a look at our high altitude sites at Glen Feshie. And again, it looks very, very similar. Just to point out again, I haven't done the statistical analysis, so we don't know whether these differences are significant statistically. But I mean, it is quite obvious that when we cross plants with Norwegian between Scotland and Norway, we completely eliminate our death rate. Like not a single Norwegian plant has died, and they've all flowered really well, even though there were a few of them, but they did quite well proportionally. So the only site that I didn't manage to squeeze onto this slide is the last site at Glen Lochy, and it looked slightly different. So got it on a separate slide here. And if we go back quickly, so the survival rates were all quite similar in the 80%, 2 years in. I don't know whether that. And then compared to that, the survival rate here was 60%. So clearly we've not used, we've not chosen the best site here. But also overall across the different treatment, it looks like there's no big difference whether your plants are crossed or not, except when we cross between Scotland and Norway again, where we have not a single plant again has died, but then also for the flowering, it's quite random across the different treatments. So we've not chosen a good site here, I think is the conclusion from that. So just to sum this up quickly, so the genetic data has shown, that the Scottish sites that we have are really inbred and have a low genetic diversity. We have less than 100 plants that remain in the UK, in Scotland. And cross-pollinations do increase plant size, survival, flowering in the nursery and in the wild, which is great. And the translocations have shown that we can grow with the plants away from the natural sites. There were some debates about that. It can be done, but grazing is an issue, no big surprise there. And our preliminary data shows that maybe if we cross plants before we plant them back out, we can actually increase our success rates for when it comes to translocations. But as mentioned, it's the long-term monitoring that's currently lacking. So this is only 2 years in, so the picture might still change just so slightly, but the initial results are actually quite promising. But yeah, long-term monitoring will be really important. So to conclude, conservation translocations and genetic rescue, particularly in combination, can be a really, really valuable tool to help plant conservation, but it is a lot of work. It is not always straightforward. It requires a lot of time, space, horticultural and scientific expertise. And ideally, and this is a really obvious point, but ideally we protect things before they become so rare and so threatened that actually they require genetic rescue or translocations, because that is very costly. We can bring them back, but it's going to be quite costly. And that was me. Thank you very much.
Thank you very much, Aline. That was absolutely fascinating, and we've caught up with time, which is fantastic. So we can now have questions for both Aline and Matt. So we've got a roving mic, and I've got some instructions here. Yes, so if you could put your hand up if you've got a question, if you could wait till the mic arrives and introduce yourself, say your name before you ask your question. That'd be fantastic. First question, and can you say who it's for as well, please?
Hi, it's Aline Grace, Deputy Director of Science, and Simon introduced me earlier, so lovely to meet you all. My question is for Matt. Thank you so much for the overview with some interesting good news as well as the more depressing trends in some of those graphs as well. We all know that plants don't observe political boundaries. So I'm curious to hear your thoughts on how the trends for Scotland are similar or different to other areas covered by the plant atlas.
Oh, thank you very much. That's an interesting question, and Pete, who's with us in the audience, is probably a much better place to give an in-depth answer. I think across Great Britain as a whole and across the political components of it, there was similarity. The overall picture was replicated that native species are losing out, and non-native species are increasing, and we now have more non-native species present in Britain and Ireland than we have native species, which is quite a remarkable thing to think about. Obviously, the proportion is perhaps a bit higher in southern England than it would be up here, but people correct me if I'm wrong, but I think the overall pictures are quite well replicated. The archaeophyte thing is quite interesting in that we seem to have done slightly worse for archaeophytes here, but I wonder if that's possibly because we have a lower proportion of cultivated and cultivatable land, so the impacts have been felt more strongly. No problem.
Thank you very much. Any questions for Aline? Okay, we've got one here in the middle, and I could see there was one at the back which we might have time for too.
Thank you, Stephen Jones from Botanic Gardens Centre. Welcome team and lapsed ecologist. I have been involved with Elaine volunteering with some certificate. Aline, did I catch you right that you've never observed seedlings of certificate in the wild, so natural regeneration? And if that's the case, I wonder if that's a feature of the Scottish flora where the plants are struggling in general, and whether or not that's a pattern that can be seen elsewhere in the plant world. Thank you.
Yeah. Stephen was also named the mountain goat. So he was particularly good at planting on some of the ledges. Sorry, I almost forgot the question there. So no, we have never seen any seedlings in the wild populations. Though I have to say it would also be quite hard to find them because to see the difference between a clonal growth or a seedling would be difficult, and we probably don't go up very often. But what we don't see is any recruitment from away from the ledges. So there could be different reasons for that. Either they don't manage to get off the ledges because they're being immediately eaten if they germinate somewhere, or because the environment has become so unsuitable that they won't be able to even germinate. So it's a bit tricky to say which one it is without looking into it more deeply. But I suspect, yes, it's not going to be the case for that species.